So hopefully this was enough time for you to ponder the answer, and the answer is B. Non-specific, the histones interact with DNA non-specifically. They can bind any part of the DNA backbone, which has phosphodiester bonds and negatively charged oxygens, which allow for an electrostatic bonding between the positively charged amino acids inside histones and the negatively charged oxygens inside the DNA. Now, right now, I would like to take a little bit of time to just look at one of these histone complexes with DNA. Hopefully, it's not going to lag. So, again, I went to PDB, which is the protein database, and this picture here is in red. You guys can see the DNA molecule, and in the green, turquoise, and light green, you can see the histones. Now let's try to rotate it. You can see that the DNA has spooled itself onto the histone twice. There's exactly two turns. One is attaching itself to one half of the histone octamer. The other is attaching to the other half of the octamer. You also hopefully can appreciate that there's things sticking out of this nucleosome complex, like this one right here, and right there, and right there. And these are the histone tails, and we're going to talk about them shortly. And the line right here that you guys can see is just, it doesn't actually exist. It's an axis showing the correct orientation of the molecule. But you can see how tightly the DNA has packaged itself, and you can also hopefully appreciate that the interaction inside the DNA, between the DNA backbone right here and the histone proteins mostly occurs through these loop structures. So actually, let's look at that a little bit further. Here are the pictures that you guys can see in your book. So this is the face view of a nucleosome, where you can see that the DNA, but you can't appreciate that the DNA wraps twice around the histone complex. And here you have a space filling model of the DNA wrapping around, sort of, and looking through the side, where you can see two DNA helixes attached to one histone. The color coding throughout this chapter of the book is very nice. The 2A is always yellow, 2B is always red, 3 is always green, and 4 is always this purplish blue. And as I mentioned just right now, there's a very particular histone fold that all of the histones have. It's this short helix, long helix, short helix with two loops, loop 1 and loop 2. If you guys look at the structure here, or if you go to PDB, you can really see the typical histone fold in these proteins. You can see that H3, that histone right there, this is the long loop, here's the short part, and here's the short part. And you can see that it is the helix, long helix, short helix, short helix. And there's this loop right here that's turned specifically towards the DNA helix. And if you go into your book, you can see that the alignment of the different histone proteins, where it is the loops that contain most of the positively charged amino acids, such as lysines and arginines. So the histone is binding to the DNA here, and here via via electrostatic interactions between the amino acids of the histone and the oxygens of the phosphodiester backbone of the DNA. Now, the long helix of the histones actually has a lot of hydrophobic amino acids, such as leucines, isoleucines, and valines, and allows for hydrophobic interactions between the histones in order to form the core of the big histone complex of the nucleosome. Another thing that I would like to point out is it's very obvious visually that this picture and this picture are very different. Here there's a lot of space and there's these nice spirals and you can see the bases. While here everything is just sort of plasticky and packed and there's no space. This is a ribbon model of a protein. This is a space-filling model of a protein. Both show exactly the same protein, but the representation is different. Why do I want to harp about it? The helices you see right there is just the backbone. Whenever you see a protein structure with just the helices and the beta sheets, you're only seeing the backbone of that particular protein. Only the carboxy amino connections of a typical protein. The side chains, the actual amino acids which make the protein fully functional, are not represented. That's why there's so much space. There's not actually space between the DNA and the histone. This is just put in here so we can visualize how the protein, how this particular fold fits into this space, because this fold too, the histone fold, does not show the side chains of amino acids, but just the backbone, which can fold either into a helix, a beta sheet, or a loop. Now the space-filling model, of this nucleosome has all the side chains present. That's why it's so full. There's nothing that you can, there's no water coming in between the histone and the DNA. But it's a lot harder to make sense of because it's so busy. That's why a ribbon model is frequently used to show a particular protein. Now, histones are not the only way to compact chromatin. To compact DNA into chromatin. The nucleosomes are called the 11 nanometer structure of chromatin, but there is higher order structures out there. They are either putative, have been observed in vitro but not in vivo, proposed, and people are still working on that. But this is a general idea of how DNA is packaged using many different levels of protein interactions, different proteins binding to each other or binding to the DNA or compacting histones and so forth, in order for us to be able to compact the DNA to 2.2 meters of DNA into a 6 micrometer nucleus. Now, there's another task that histones also perform, and this is DNA organization. Remember from the beginning of this lecture, I talked about how it's not just important to stuff the DNA into the nucleus, but it's also important to be able to transcribe something from it. If you can't make RNA off of it, why do you need the DNA? You can't run a cell. So how is it that the DNA molecule that's packed into histones still remains accessible to mRNA transcription? Now, we already briefly mentioned that in euchromatin, so a more open chromatin, the nucleosomes are not as tightly packed, while in heterochromatin, they are much more closer together, preventing access from DNA polymerase or any other protein, really. So how do the nucleosomes know to pack loosely or tightly? Well, histones can be modified, and they're modified on these tails we just saw that are sticking out of the nucleosome and can be readily bound by other enzymes. And the histone modification, the modification of the tails of the histone will either allow transcription or force gene silencing, depending on what kind of modification it is. So here you go. You guys can see these things protruding from a nucleosomes. When we looked at them in PDB, they were just little lines because only the backbone of the N-terminal histone tails was shown. Here, this is a space-filling model that has all kinds of, that has the side chains as well. So that's why you see bumps and circles and so forth, because these histone tails also contain a lot of lysines and arginines, as well as serines and all kinds of other amino acids that are can be readily modified by different proteins. The process of modifying a protein after it has been translated and folded is called post-translational modification. You can apply that. This term can be applied to histones, but all kinds of other proteins as well. And what I would like to say for you guys to actually appreciate what X-ray crystallography can or cannot show us is that this is how the histone tails might look. But because they're so flexible and mobile, you can actually not fix them inside a crystal structure. So they're not ordered enough in order to be crystallized. Because in a protein crystals, you can't have any motion. Every atom inside a protein crystal has to be fixed in one particular location and orientation so that you could interpret the diffraction patterns from a protein crystals and then pinpoint where each individual atom is located. So you guys can see the front view and the side view of these histone tails that are protruding from the histones. And because what histone tails can do is they can be post-translationally modified, and depending on the level or type of the post-translational modification, they can either bind with each other, further compacting the nucleosomes into the snarl, or they can repel each other and instead interact with water, allowing for more loose nucleosome arrangement in the DNA and giving the RNA polymerases or transcription factors a chance to get in there and make some RNA. So this is basically what this blurb says. So there's lots of different post-translational modification that have been reported in the literature. This is just a brief summary, and what I would like for you guys, there's two specific post-translational modifications into very particular amino acids we're going to discuss a little bit later, and those are worth remembering because we're very sure about those. However, trying to pretend that this code, that there is an actual code existing that will make sure that every single methylation does this thing and every single acetylation does that thing, that's not true, and there's still a lot of research being done on the subject. So let's look at them at a little bit more detail. So you guys can see here that all kinds of histone tails, and it's usually the N-terminal tail, though some histones have a C-terminal tail as well. It's mostly the N-terminal tails are modified, and you can have methylation, either on an arginine or a lysine, acetylation, typically on a lysine, but also phosphorylation on a serine or ubiquitination. Here, you can see the model of a nucleosome with the tails protruding. Here, this is just the, let's say, primary structure represented as a line of each individual histone. Again, you don't need to memorize it, but appreciate it that all the histones have an HFD, which is the histone fold domain. Remember, short helix, long helix, short helix. And the HFDs are less modified, but it's mostly the tails that contain modifications. H3 tails is a really long dude, so it's frequently modified. In this case, you guys can probably appreciate that there's lots of different colorful dots here. Well, there's only two types of dots here because this diagram just shows the places where different histone tails can be methylated, and that's it, and phosphorylated, of course. So this is the summary figure from your guys' book, and there's many reviews on the subject as well, published yearly, that sort of summarizes the different methylation, the different modifications you can find just on one particular tail, the tail of the histone 3, or the histone 4, or the SNP-A, which is sort of a buddy of histone 3 and 4 and plays a role, is a type of histone attached to the nucleosomes when the cell plans on dividing through mitosis. You can see the vast different types of different modifications that can occur, and based on the amino acid position, it can have a totally different effect. So this thing is an acetylation. You can see that this acetylation modification is slightly more N-terminal than this one, and because it's on one amino acid and this is on the other amino acid, the effect is very different. This one allows for transcription to occur, and this one, in fact, recruits more histones to the location. Crazy, right? So it's a hot topic of research. A lot of work is still being done on it because the way histones affect gene regulation is very important to understand, both for cancer, for a lot of other diseases such as diabetes and Alzheimer, and just understanding epigenetic inheritance, you have to understand different histone modifications. And you cannot really call rules on most of them. However, there's three locations that we can understand pretty well. So the modification on histone 3, lysine 9, is one of these locations. If you methylate H3K9, this causes transcriptional repression. If you acetylate H3K9 and instead methylate H3K4, that causes transcriptional activation. And these methylation, demethylation, acetylation, deacetylation is done by these chromatin modifying enzymes. They're enzymes because they either attach a methyl group or remove it. Who knew that a simple methyl group would have such a giant effect? Also, I want to, again, you can read it, but I want to spell out the importance of notation. So in this histone methylation areas, because different amino acids will have a different effect, I usually refer to as H3K9, H3K9 met, H3K9 acyl, or whatever. That means that the histone altered here is histone 3, and it's the lysine which one letter amino acid abbreviation is K. It's the lysine in the number 9 position if you count from the starting methionine, from the N-terminal methionine of the protein. So what happens when these methylations occur? Acetylation, specifically on K9, opens up the chromatin. It's a tag that allows other things to come in and then start RNA transcription from that area, while methylation on K9 closes it up. And multiple enzymes are involved in the process. There's a deacetylase and a putative demethylase as well. You can't just remove the acetyl group in order to methylate. You also have to add a methyl group. So there's also histone H3 methyltransferase. And you guys can see that the process is reversible. You can methylate, acetylate, demethylate histone all day long. It doesn't affect the histone core. So histone modifications are reversible. So being able to find these enzymes that can do deacetylation or methylation of a particular histone area will allow you to affect the epigenetic state of a cell. So a lot of research has been done on the subject. And if you guys care, I would recommend reading the intro paragraph of the chapter 10 that describes a scientist from Rochester, New York, actually describing the process of how they isolated the first HAT, which is a histone acetyltransferase.